Okay, so let's continue our lesson on DNA. We're going to talk about protein synthesis today. Your objective is to be able to explain how DNA and RNA code for proteins and determine traits. So protein synthesis is the process of reading the instructions in the DNA to make a protein. We're making proteins. We call protein synthesis the central dogma of genetics. That means that we go from DNA to RNA and from RNA to a protein. DNA are the instructions and it is in the nucleus, but it can't leave the nucleus. So, but the protein is made on the ribosomes. So protein synthesis takes two steps. We go from DNA to RNA. We call that first step transcription. And from RNA to a protein, we call that second step translation. So just as a review, look over this chart of the differences between DNA and RNA. So we have three essential types of RNA. We have messenger RNA. That copies the instructions in DNA. Messenger RNA copies the instructions in DNA, and it carries these instructions to the ribosome in the cytoplasm. tRNA, or transfer RNA, it carries the amino acid. This is the amino acid. This is the attachment site for tRNA. It carries the amino acid to the ribosome and the messenger RNA. And these are ribosomes. Ribosomal RNA is the ribosome itself. You rarely hear me say ribosomal RNA. I just tend to say ribosome. So transcription, the purpose is to carry the code or instructions out of the nucleus. Excuse me. Remember, DNA never leaves the nucleus, and proteins are made in the cytoplasm by the ribosomes. So transcription carries the code, the instructions out of the nucleus. Transcription, this whole process is only located in the nucleus. Transcription starts with DNA and it ends with messenger RNA, but it's only the process that takes place in the nucleus. And there, DNA is copied into a complementary strand of messenger RNA. When free-floating nucleotides form the messenger RNA in the nucleus, the base pairs are different. So up to now, we've been learning more so about DNA, the complementary bases. Well, recall that RNA does not have thymine. It only has uracil. So if the original DNA strand has a T, T is still complementary to A. So the messenger RNA can have A. But A, for the original DNA strand, the complementary base is a U. Why? Because RNA does not have thymine. So the process of transcription is to unzip the gene that needs to be copied. We use the complementary base pairing rules to match RNA nucleotides with the exposed DNA nucleotides. So this purple part is DNA, right? DNA has been unzipped. The red is the messenger RNA. Notice how the red, the messenger RNA, has the uracil on it. We're still using the complementary base pairing rules, but remember, it's uracil instead of thymine. And then the last part of transcription is to release the completed messenger RNA molecule from DNA. DNA zips back up, and then the messenger RNA is allowed to leave the nucleus. So again, transcription, DNA is copied into a complementary strand of messenger RNA. So this is DNA unzipped, right? The hydrogen bond separates the molecules, and then the messenger RNA uses its complementary base pairing rules to match up the bases from DNA. So let's try it. Let's practice a little. So if this is a DNA sequence, let's look at the first three letters, TAC. What would the So if the DNA sequence is TAC, let's just look at the first three letters, right? The messenger RNA sequence, what goes with T? What goes with A? What goes with C? Yes, it would be AUG. Remember, RNA, regardless of the type of RNA, it does not have thymine. The second step in protein synthesis is called translation. The purpose is to read or follow the instructions carried on the messenger RNA to make a protein. It assembles the amino acids in order. So translation is located in the cytoplasm, specifically on the ribosome. We start with messenger RNA and we end up with a protein. Okay, so again, let's go through the steps of translation. The first step of translation is for the messenger RNA. And remember, this is the second step in protein synthesis. The messenger RNA attaches to a ribosome. The ribosome, rRNA, reads the messenger RNA codon. A codon is 3 consecutive bases on the RNA molecule. It's like a triplet. We'll get some practice with that in a minute. The transfer RNA, tRNA molecules, they act like a taxi. They pick up and they drop off the amino acid that match with the current codon. And we're going to go through all of this. I'm going to take you through every single step in like an illustration. The fourth step is for tRNA to continue to drop off the amino acids as the messenger RNA moves through the ribosome. So if you would imagine that the ribosome is like one of those check cashing cards at Food Lion or Walmart, and the messenger RNA is like your debit card. So the messenger RNA moves through that machine just like your card would read all the information from that machine. The ribosome binds the amino acid together with peptide bonds. So far we learned about 3 different types of bond. Hydrogen bond is a weak bond, covalent bond is a strong bond, and a peptide bond is the type of bond between the amino acids. When we reach the stop codon, and I'm going to go over that, the ribosome releases the completed protein. That means the process is done. So translation, again, we're interpreting the RNA message into a protein. Notice how the red is the messenger RNA. This is the tRNA. Is carrying the amino acids and is bonded to the base pairs, A to U, U to A, C to G, and G to C. So how is the code read? Messenger RNA is read 3 consecutive bases at a time. The bases are only on messenger RNA. We call it a codon. 1 codon is equal to 1 amino acid. When you have many amino acids bonded together, they make a protein. We have 64 codons and 20 amino acids. I'm going to go over the chart with you. So this is tRNA, right? tRNA is called the anticodon. This one is the messenger RNA, the red one, that's the codon. 3 consecutive bases. So here's the vocabulary for protein synthesis. The genetic code is a code of instructions for how to make those proteins. The codon is a set of 3 consecutive nucleotides on the messenger RNA, and of course I want you to draw that. The anticodon is 3 consecutive bases on the tRNA, and the amino acid is on the tRNA at this attachment site. Pause this video if you need to. So this is the universal genetic code chart. I want you to highlight, rewrite. I just want to make sure you know that this chart, you're only looking at the messenger RNA codon. You're only looking at the messenger RNA. So this is the first base on that messenger RNA. This is the second base, and this is the third base. So if we were to look at the messenger RNA codon AUG, A would be the first base. So we have narrowed it down to somewhere in this row. U is the second base. We've narrowed it down to this box. And you take your finger to G, take it across the page. AUG is the start codon. So now you're going to have some practice with it. There's the DNA sequence. Again, if it starts, the DNA sequence is TAC. The messenger RNA sequence would be AUG. The tRNA sequence would be UAC. The codons are the 3 consecutive bases at a time. And looking at your amino acid chart or your messenger RNA chart, you would get your amino acids. Okay, so let's put it all together. So this is a cell, right? In this cell, you have, this is the nucleus right here, these 2 lines. That's the nuclear envelope. Everything that occurs inside the nucleus is called transcription. DNA unwinds, the messenger RNA transcribes the message. Messenger RNA leaves through the nuclear pore. It goes into the cytoplasm where it finds a ribosome. The messenger RNA translates the message to the ribosome, just like your debit card going through one of those machines. tRNA, that's your taxi. It brings that amino acid over to the ribosome machine. As you stack up, as the amino acids stack up together, you have a polypeptide chain. Once it finishes all of the amino acids that it needs to bring, we call that a protein. Oh, and one more thing. These 3 bases on the tRNA is called the anticodon, and these 3 bases on the messenger RNA are called the codon.